Good morning, and I'd like to thank the organizers for the opportunity to speak here, especially in such a fantastic symposium. And I thought that the three previous talks are just fantastic in terms of explaining a lot of the progress that has been made in terms of understanding genetic hearing loss and trying to be able to find strategies to change things. So I'm going to switch gears a little bit, and I'm going to talk to you about some of the work that my lab has done in cochlear regeneration, which is a little bit different from talking strictly about genetic models of hearing loss. And that's because acquired hearing loss in adults can come from a number of different causes. So we're all aware that chemotherapy or certain antibiotics can cause hearing loss. That noise exposure is a large contributor to hearing loss, and also that certain diseases such as HIV, mumps, measles, and a number of pathogens that affect small children can also cause hearing loss. And so my laboratory is focused on chemotherapy, antibiotics, and noise exposure as causes of hearing loss, and that's what we're trying to treat. So I think we're all aware that mammals don't regenerate lost cochlear cells. And so the picture that I have on the left shows you an image of a guinea pig cochlea, and you can see the 3 rows of outer hair cells and the single row of inner hair cells. It's a beautiful structure. But on the right, you can see a cochlea from an animal that was treated with antibiotics, and you can see that the hair cells have been lost. So there's scar tissue, and in that region, there's not going to be any amplification of sound. And so it's going to be very difficult for the ear to detect sound. And this contrasts with non-mammalian vertebrates like birds. So I'm showing you a picture of the bird cochlea here on the left, a normal bird cochlea. You can see hair cells there, just like in the mammal. And if we treat them with antibiotics similarly, you can see that in 3 days, all of the hair cells are gone. But if you wait about a month, what we find is that the bird will actually regenerate these hair cells. Now, this was discovered by 2 groups and published simultaneously 30 years ago in the journal Science. So we have known about this phenomenon for 3 decades. One of the things that you really that was important to show afterward, once we knew that the cells were there, was to show whether or not those cells would work. And so in a very important paper by Brenda Reels, who was one of the original discoverers of regeneration, she showed that a bird that loses its hearing from antibiotics injection loses that hearing, and they recover full functionality. They're able to discriminate between songs when they recover their hearing just the same as they did before their hearing was lost. And this is really important because this tells us that not only are the cells able to recover, but that the function follows with it. And so this is one of the things that guides me. This is what I want to really understand. I want to know if this is possible to do in humans. And if it is possible, how can we make it happen? And I'd like to point out that in birds, the function is observed 40 days after the loss of hearing. So it takes time. It takes over a month before the birds are able to hear again. And so we kind of conceptualize this problem when we talk about it. We have hair cells in yellow. We have supporting cells in blue. If we induce damage, then the hair cells are lost. And what we imagine is that somehow activates the supporting cells. There's either a signal that is provided or there's a loss of a signal that's provided to activate these supporting cells. And when they're activated, it's been shown that they divide, some of them divide, and turn into hair cells, and some of them just directly turn into hair cells. So both processes are observed in the bird. And so the process where there's cell division, we call mitotic regeneration. And the process where there isn't any division, where the cell just transforms itself into a hair cell, we call that direct transdifferentiation, because you go from a cell that is fully differentiated as a supporting cell, and it becomes a completely new cell type. So a number of years ago, I was part of a team that conceptualized this problem by saying maybe the issue is that either the cells don't have the ability to do this, that they don't have the ability to become new hair cells, or maybe that the signal is absent. So that kind of gets back to here with this idea that damage is going to cause a signal, and then the supporting cells are going to be activated. So because this doesn't happen in mammals, we thought either the signal doesn't occur or the cells don't have the ability. One of those two things, or both of those things, is going to have to be true. And so in order to test this hypothesis, whether or not the cells have the ability, what we did is we developed a way of purifying supporting cells. So what you see on the left in the top panel in red, you can see some staining which shows you two lines, and those are lines that are identifying specific cells in the cochlea. The hair cells are in green, and they're in the second panel just below that. And you can see in cross-section in the bottom panel, there's green hair cells, and then there's red cells surrounding them. Those are the supporting cells. And we used a technology called a cell sorter to purify those supporting cells and place them in culture. And what we could do is we could separate the hair cells, which were green, and those in the dot plot, the scatter plot that you see there in the middle. The green cells are in the lower right quadrant, whereas the supporting cells are in the upper left quadrant. So we could separate them completely, and we could place them in culture. And you see a picture of one of those cultures there on the right-hand side. You see the cells in gray, and if you look in the green channel, you don't see any hair cells at all. And when we waited a week and we looked again, we found new hair cells had differentiated. And so this was evidence that those supporting cells still had this ability. They could make new hair cells. They could at least start the differentiation pathway, even though this is not something you ever see in an animal. And when we looked at different markers, further differentiation markers for hair cells, we found that they were able to do that. So the top row shows you that they made little stereocilia. That's those little red triangles in the middle panel. And then the bottom row shows you that they had functional hearing channels. FM143 is a dye that the hair cells take up when they're electrically active. So the key thing for this paper was that we were able to show that this potential continues even after hearing begins in the animal. So in mice, like in kittens and puppies and a number of other animals, they're born blind and deaf. And when we first took the supporting cells out of the mouse, we took it out of a mouse before it had begun to hear. They were only a few days old. And so this was a problem because human babies develop the ability to hear at around 8 months gestation. And so we were looking basically at what was the equivalent of a human fetus. So what we wanted to do in this study was we looked at animals that were older than that. We looked at animals that were 14 days old. They were about halfway between birth and sexual maturity. So when we took supporting cells from those cochlea, and you can see a diagram of the cochlea in cross-section with the hair cells labeled in red. And below it, you can see the section of the transgenic animal showing you the green cells that we isolated. We found that those juvenile supporting cells were also able to make hair cells. So that suggested that the potential was maintained, at least into early childhood, for the ability for these supporting cells to become new hair cells. So those results were we were the first ones to publish that in the study, but they have been repeated and they have been extended in hundreds of reports from dozens of laboratories. And people have shown that in the damaged mouse at the newborn stage, there is spontaneous hair cell differentiation. And people have extended and identified further markers and characterized these stem-like cells. And people have also experimented with turning on hair cell genes and turning off supporting cell genes by introducing factors that alter the expression of different proteins in the cells. And people have also experimented looking at DNA accessibility for hair cell genes in trying to understand how to promote this ability to drive proliferation. And people also are analyzing candidate signaling pathways, trying to figure out how we could drive this, this phenomenon to happen. And that's really the big question. It's been 30 years since we discovered that birds are able to regenerate, and we still don't know what the signaling pathway is that is able to activate supporting cells. This is so hard to find something that you don't know. And so I'm going to talk a little bit about the work that I began when I started my own laboratory on working on candidate signaling pathways. And I just wanted to give you, in the little diagram, you can see a very crude cell. And there's a receptor that's shown in blue. That's a protein that's on the surface of the cell. And in yellow, there's a little circle, and that's called a ligand. And we can call it a ligand, or we can call it a signaling factor. But what it means is it's going to be something from outside the cell that signals through a receptor pathway. And that receptor pathway is going to activate, change things, change the cell itself. And it's going to be passed through the cell, and the cell is going to mount a response. So that's the idea behind this, is the thought that there could be such a factor, a ligand, a signaling pathway, or a combination of them, that can act on a receptor and drive differentiation of hair cells. That's what we want. And people have studied a number of pathways, and I know that this is sort of a gamish that we call one pathway Notch, we call one pathway Wnt, and we spell it W-N-T. There's another pathway people are studying, which, I'm not joking, is literally called Sonic Hedgehog, and it was named after the character in the video game. At least you're not going to forget that one. I study a receptor family that's called the ERB-B family. So that's the last one there. Now, ERB-B is a controversial candidate for hearing restoration. And that's because the ERB-B gene is responsible for a number of human cancers. So when this gene is activated in places where it's not supposed to be activated, it can drive breast cancer and it can drive schwannomas, which can also disrupt hearing. And so studying the ERB-B receptor pathway is driven by what we've found so far, and what we're trying to understand is how we could use the things we've learned to develop therapies. But I want to caution you that we also have to take into consideration the fact that this receptor pathway is very powerful, and activating it in inappropriate places can be very dangerous. And so we have to understand what the downstream factors and pathways that it activates might be. Those would be things that we would probably target. But this is trying to understand regeneration from the top down. So one of the reasons why I think that ERB-B is an important one to consider is because some early studies that we did in collaboration with Jenny Stone of the University of Washington was to look at chicken supporting cells and to see whether or not interfering with the ERB-B pathway could block regeneration activities. And so this was one of my first papers as a PI. And what we found was that if we used an inhibitor, and the name, so there's a timeline there on the left, and you can see 0, 24, 48, 72 hours. This is describing the culture of the chicken cochlea once it's been put into culture. So it goes into culture, and at the very beginning, we add an ototoxin. It was streptomycin that killed off hair cells. And then we waited one day for the hair cells to die, and we added a chemical that could block ERB-B signaling. And that chemical is named AG1478. We waited another day, and then we added a third chemical that allows us to label cells that are going to go into division. So cells that are going to proliferate have to duplicate their DNA, so they have to incorporate in molecules to make that DNA. And one of the things we can do is we can add in molecules that are kind of like DNA building blocks, but not exactly, and then look to see those later. And that's what BRDU is. It's a chemical that gets incorporated into DNA, and then it persists as long as the cell lives. And then we would fix the cultures at 72 hours and count for BRDU positive cells. And I didn't show a picture of what that looks like, but on the right, you can see a graph that shows you the quantification of BRDU positive cells. And in the absence of any AG1478, we get a lot of proliferation. But as we add more and more of it, you can see that the proliferation drops down to about 25% of the original level. And so this shows you that ERB-B is definitely involved in the natural regeneration pathway. And this is neat because we know that ERB-B receptors and ERB-B signaling molecules regulate regeneration in other epithelial tissues. So the image on the left is an image from a trachea, human tracheal cells, and it's just showing you a single layer of the cells. And the green labeling on that is showing you the ligand, the signaling molecule for this pathway. And that's anchored at the top of the cells. And at the bottom of the cells themselves in the epithelium, there are the receptors for that ligand. So the trachea epithelium carries with it the ability to regenerate. It has both the signal it needs and the receptor that it needs. And it's only in cases of damage that the ligand is able to contact the receptor. If there's a stretch damage or if there's a tear damage, then that signal, the regeneration signal, is right there, and it's able to bind to the receptors and drive proliferation. And this is true in a lot of epithelial cell types. What's really interesting is that the receptor, ERB-B2, is present in the adult mammal cochlea throughout life. It's always there. And the ligand, the signaling molecule, which is in green, it's named neuregulin, it is also present in the cochlea. And just like in the tracheal cells, it's anchored at the top of the cells, whereas the receptors are all along the length of the cells. And so the presence of the ligand and receptor are there in the cochlea, just like they are in regenerating tissues, but they don't function to drive regeneration as far as we know. And that's really the big question. So if the supporting cells can make hair cells and the pathway proteins are present, what's stopping it? And that's a great question. And so we thought maybe the signal is blocked. So the way to try to do this is to use modern genetic tools, and this is all done in mouse. We get mice that are genetically modified to carry receptors that are always active. So in this mouse model, we have what we call a constitutively active or an always-on version of ERB-B, and we can use different genetic tools to drive the expression of this in the cells that we're interested in. And we're interested in supporting cells. And so we bred together mice to be able to make this happen. And the way that the mutation works is that there is a change in a protein, an amino acid, in the transmembrane region for this receptor. So then the receptors will aggregate together and turn themselves on. And that's what I show in that diagram on the left. In the middle, you can see green and red cells, and this is in a mouse cochlea that contains all the transgenes to turn it on. And the red cells is the active protein, and the green cells is a lineage tracer that we use to be able to identify and follow those cells later. And on the right, you can see that there's no such staining in the controls. So when we did this, basically when we did the study, one of the first things we wanted to look at was supporting cell markers. We wanted to know if the supporting cell markers stayed on in a mouse where we were able to activate this gene. And this is what I'm showing in this figure. So what it shows you on the top is the control. So the panel on the left is red staining, and the control doesn't have any because it doesn't have the active gene. And in the middle, you can see the supporting cells marked in cyan. And so you can see the supporting cell markers are all there, and they look pretty good. But on the bottom three panels, There is the phospho-ERB staining, so you can see we were able to activate the gene. And one of the things that it did was it downregulated the supporting cell marker. So that suggests that this may actually function in part of the regeneration pathway. Because to make supporting cells into hair cells, they have to both turn off supporting cell genes and turn on hair cell genes. And so we looked for new hair cells in these cochlea, and so we let the mice live for a number of days because we knew that it would take some time for those hair cells to develop. And we didn't see as many hair cells as we might have expected given the downregulation of the supporting cell marker. But we did find them. So in the top panel, which is the control, you can see the hair cell markers in red and purple, and the cell that would be the ERB positive cell in green. And you can see the pattern is normal in the top, but in the bottom where we've activated ERB, we have scattered little new hair cells in the supporting cell layer. So those are those rounded purple cells that are lit up with the yellow arrow. So these are new hair cells present in there. But the weird thing is these hair cells are not the cells that expressed ERB. So the cells that are signaled, the cells that are expressing ERB, are somehow telling their neighbors that there's damage and it's time to make new hair cells. So this was really surprising to us, and it took us a long time to understand this. But what it suggests is that the pathway for regeneration is a multi-step process. And if ERB is involved, this is evidence, but it's not proof. If ERB is involved, then ERB is toward the top, and it is activating other cell pathways, a dialogue between the cells to drive this kind of differentiation. So we have ongoing experiments. We're testing ERB activation in adult mice after damage. So we are looking at both noise damage and drug damage. And I don't have results to show you for this today because these experiments are ongoing and we don't want to publish something that we can't really be absolutely certain that we're sure of. And you may be thinking about this and you say, this is really interesting, but it doesn't explain to us why in mammals the signal is blocked. Why would that be? Why would it be that the signal is there and the cells are competent, but everything is blocked? And the answer is that people have actually studied the function of ERB, what it's really doing in the cochlea in normal conditions. And one of the things that they found is that ERB in the normal conditions seems to be part of a feedback signal that promotes the maintenance of synapses. And so ERB expressed in supporting cells, which are not shown here, helps the hair cells to secrete signals for the neurons. So neurons are shown in yellow. The neurons have come up and their projections touch the hair cells, and their projections allow for the signal to go from the hair cell to the supporting cell. And this involves a complicated pathway of factors that the hair cells express that allow the neurons to maintain these connections. And so what people think that ERB is doing normally in the cochlea is helping to maintain connections, which is an important factor, an important thing to know. What our experiments do is they drive ERB signaling down a different pathway within the cell itself. So this signal would be presumably going down one set of second messenger pathways downstream of ERB, whereas our signal would be going down a different set, which is somehow inhibited. And so that's where we're trying to focus our efforts to understand if we can lift that inhibition just even for a brief while, maybe we can drive some form of restoration. So that's the work that my little lab is doing. There are several dozen laboratories that are focused on cochlear regeneration and hearing restoration, and they are taking complementary approaches to the ones that I'm showing you here. So we're all looking at the idea of how do supporting cells turn into hair cells, and some people are working with drug combinations to try to activate those supporting cells and turn them into hair cells. And other people are looking at different proteins that are inside supporting cells or that they can introduce into supporting cells to try to drive hair cell differentiation. And it's not just the academic world that is studying this. There are over 40 companies now that are funded and that are looking at different ways to try to drive hearing restoration. And there's things that we have to try to better understand. So one of them has to do with the changes in the abilities of supporting cells between newborns, juveniles, and adults, and whether or not the abilities that are present in the newborn and the juvenile continue to be active in adult supporting cells. And another issue that you should always look for are questions about cochleas that are taken and placed in culture and experiments that are done in culture versus ones that are done in the animal, which we call in vivo, because these are very different sets of circumstances for the cells. And sometimes cells in culture are able to do things that they can't do in the animal. And so all of the experiments that we do in culture, we then have to replicate with animal studies. And I'd like to thank my students at the University of Rochester here in Rochester. I hope you're enjoying the city here. And my colleagues at Rochester, as well as my collaborators at the Massachusetts Ear and Eye Institute and our funding sources. And thank you for your attention.